Let's focus on the somatosensory system, which is used to sense touch, pain, temperature, and proprioception, which is in the muscles. Let's look at this practice question. Touch activates mechanoreceptors. Those are the sensory neurons for touch. What is the likely response in mechanoreceptors during a touch stimulus? So the key word here is activate. So you're looking for something that's going to cause action potentials in the mechanoreceptors. So the best answer here is sodium channels opening, right? Because sodium will go into the cell, cause a depolarizing graded potential, and maybe get the cell to threshold for an action potential. So the way this works is that in the membrane of the dendrites, in the mechanoreceptors, there are these special ion channels that are permeable to sodium, and at rest, they're closed. These channels are gated by a mechanical deforming of the membrane. So the membrane has to change shape in order for these ion channels to open. So these are called mechanically gated ion channels because they're gated by a mechanical change. And these ones are permeable to sodium, as I said. So when there's a touch stimulus, it's going to cause the channels to open, right? So the touch is going to, let's put it here. So here's our touch stimulus onto the skin. Right underneath the skin are the dendrites for this neuron. And when the skin gets pushed on, then the cell, the dendrites, will actually get pushed inward as well, right? So the cell membrane is changing its shape because of the mechanical stimulation. Something like that. Okay, so it's the same cell, same dendrites, but when there's a touch on the skin, it pushes in the membrane. And so that causes these channels to open, right? So they open, and as we just said, they're permeable to sodium, so sodium's going to rush into the cell and basically cause graded potential. Now, it doesn't necessarily mean that it will trigger an action potential. This touch stimulus needs to be strong enough that enough of these mechanically gated channels will open and enough sodium comes in to cause a large enough graded potential so that we get an action potential. So we'll say, maybe there's an action potential. If there's a touch stimulus but no action potential is triggered, then you actually will never be consciously aware of that stimulus. Now let's look at thermoreceptors, which can sense temperature. So there are some thermoreceptor cells that are specialized for sensing warm temperature, and there's others that are specialized for sensing cold temperature. So let's say that we have a warm thermoreceptor, and this is in the dendrites. And these would also be found in the skin. Okay, so this is our warm thermoreceptor. So in the membrane, there are temperature-sensitive channels. These are called TRP, or trip channels. And basically, they're temperature-sensitive. Or temperature-gated is another way of saying that. Right, so these are closed at rest. And when there's heat and the cell gets warmed up and the channel gets warmed up, it causes the channel to change shape and open. And these are also sodium channels. So sodium is going to go into the cell and cause a graded potential. Right? And if it's strong enough graded potential, then that causes an action potential. And then that gets sent to the brain, and your brain interprets that as something that's warm or hot. Right? So again, if that action potential gets to the cerebral cortex, to the primary somatosensory cortex, then you will perceive it as hot. One thing that's interesting about these temperature channels, the trip channels, is that they're also gated by chemicals. Right? So there's an agonist that binds to this trip channel, and it's called capsaicin. Capsaicin is found in hot peppers, things that are spicy. And so when this chemical binds, even if the temperature is normal or cold, the gate will open up and the positive ions will go in and we'll get our graded potential. And then that will cause an action potential if it's strong enough, right? And that action potential goes to the brain. And how do we interpret that? We interpret it as hot, right? Because this neuron, which normally sends signals about warm temperatures, is active and it's sending a signal to the brain, and so the brain goes, oh, okay, so we are warm, we have a hot temperature here, when in actuality, it's just a chemical that's binding to the channel. So that's why you might feel warm or hot when you're eating these spicy foods. It turns out there's a number of different trip channels. The names are shown here. You don't need to really know them. But this shows the temperature when they're active. And this is in Celsius. So down here, these are channels that are active when it's cold. So they would be found in the cold thermoreceptors. And what's interesting is that they're also activated by their own agonists. Like mint, for example, might have kind of a cooling effect because it's activating one of these cool trip channels. And then we can see the chilies, we just went over that, and that activates one of the warm trips, and then we have some that are for very, very hot temperatures. Okay, so once we've activated the somatosensory receptors, the sensory neurons, then that information gets sent up into the spinal cord and then to the brain. So which lobe is the primary somatosensory cortex in? This is where your information about touch, temperature, and pain is going to go in the cerebral cortex. So the answer is B, that's the parietal lobe. So kind of right towards the front of the parietal lobe is where we find the primary somatosensory cortex. So we'll talk more about that in the next slides. This slide shows the actual pathway from the periphery, from the sensory neuron to the primary somatosensory cortex, right? So we're seeing slices through the brain here. This is the primary somatosensory cortex. This is a cross-section through the medulla, and then cross-section through the spinal cord here. And so touch neurons and also proprioceptors, which we'll talk about later, they both follow the same pathway to the somatosensory cortex. So let's say that we are sensing touch on the right side of the body. So usually this is kind of drawn in the opposite of how you're seeing it here, where that's the right side and this is the left side. And so you are sensing touch on the right side here. This one gets activated. Its neuron goes into the spinal cord, and the axon goes all the way up the spinal cord to the medulla, right? Like the first synapse is happening here in the medulla oblongata in the brainstem. I just want to point out one thing here, which is this guy. So that is a cell body. And remember that the cell bodies of somatosensory neurons are in a structure called the dorsal root ganglion. We talked about that before when we went over the anatomy of the spinal cord. Okay, so that first neuron goes all the way up the spinal cord into the brain. Then it makes a synapse. And so the second neuron, we call this the second order neuron. It crosses over to the opposite side. And then it goes up to the thalamus and makes a synapse. And then the third order neuron goes from the thalamus to the primary somatosensory cortex. So that's for the touch pathway. Pain and temperature neurons, they follow a slightly different pathway. So they come in, again, here's the dorsal root ganglion. They make a synapse right away in the spinal cord. The second order neuron crosses over and goes all the way up the spinal cord through the medulla to the pons, to the thalamus. And then there it makes a synapse onto our third order neuron, which then goes to the primary somatosensory cortex. So either way, whichever pathway you take, information from the right side of the body is activating the left primary somatosensory cortex, and information from the left side of the body is going up to the right primary somatosensory cortex. So you always get this crossing over happening. The only difference really between these two pathways is the path of the first neuron, right? So in the case of touch, it goes all the way up to the medulla. In the case of pain and temperature, it makes a synapse right away in the spinal cord. So now let's take a look at the primary somatosensory cortex in the parietal lobe. So this is sometimes known as S1 for primary somatosensory. And what's interesting about it is that there's a map of the body on the primary somatosensory cortex. So what I mean by that is that touch information about the legs is going to activate this area of the cortex. Touch information about the arm is going to activate this area of the cortex. About the head is going to go there, and so on. Right? So it's kind of in the map of the human body. This is known as the somatotopic map. What's interesting is that you could, like during brain surgery, you could stimulate this area of the somatosensory cortex, and the person who's actually awake during the surgery would think that they're being touched on their elbow. Right? So all you have to do is activate these neurons and you perceive touch or pain or temperature. Another thing to note about this is that the area devoted to each part of the body is not even, right? So this is not a properly proportioned person that we're seeing here. So there's kind of a pattern going on here where the areas of the body where we have very fine discrimination, like where we can sense things very well. This would be the fingertips, definitely, right? We're always touching things with the fingers. And then also the face and the mouth. So we have very fine discrimination there. The sensory neurons in those areas cover a smaller area, so their dendrites don't branch as much. We call this a receptive field, so they have a smaller receptive field. So they're like devoted to a smaller area of the skin, which also means that you can pack more of those sensory neurons into that area. So there's a higher density of the sensory receptors or sensory neurons. Okay, and so then since we have so many neurons there, we're going to need a larger area in the somatosensory cortex devoted to that part of the body, right? So you have an increased area in S1. Oops. Okay, so that's the pattern there. So these areas where we can do very fine discrimination, like the face and the fingers, we can see that they have a huge amount of cortex devoted to them, like almost a third or maybe even half of all the sensory cortex is devoted to just the hands and the face. So we can do very fine discrimination there.